NIDs also. So
Okay.
every university.
So these are the people, otherwise if it had not been a lockdown, would have gone for Shodh Yatra, is it?
No, exactly not, ma'am. This is the hands-on training national workshop in collaboration with the BIIS. We are organizing every year. We are organizing 3 such workshop every year. We invite the students from all over the India for 21 days, and we provide some ideas and some grassroots innovations or traditional practices to validate in our lab. So this way we train the students, and mainly the focus is to invite the aspirational district students, and then after tier 1 and 2 and 3 students.
Okay. Okay.
But due to pandemic, this time we are organizing the webinar, so later on we'll invite them for the hands-on training.
Okay. So Dr. Anil Gupta also joins once a while for the workshop?
Yes, ma'am.
Okay. So there are 39 participants, is it? How many participants are attending this workshop?
There are 60, ma'am, but right now I don't know. They have some of they have exams and clash in the college classes, and so
okay.
there fluctuation every day. Yes. So students, shall we start? All back? Ma'am, I'll take 1 or 2 minutes to give a brief introduction to the students.
Sure.
So students, we have with us Dr. Mitali Mukerji, is a Chief Scientist at CSIR Institute of Genomics and Integrative Biology, Delhi. She did her PhD in Bacterial Molecular Genetics from Indian Institute of Science in Bangalore. She works in broad area of genomics and molecular medicine. She has been the convener of Indian Genome Variation Consortium, which provide the first comprehensive genetic landscape of the Indian population from disease genomics perspective. Her group has also demonstrated how this basal data can be used for dissecting disease genes, identifying signature of selection, tracing mutational histories, and also for pharmacogenomics studies. Her group has been involved in genomic studies on spinocerebellar ataxia, a rare neurodegenerative disorder, for the last 20 years in collaboration with the AIMS, and have identified novel mutations and founders for the disease in Indian population. Her group has pioneered the area of Ayurgenomics, which has provided the first evidence for molecular correlates of Prakriti that leads to identification of predictive biomarkers for high altitude adaptation. A Trishutra Ayurgenomic Consortium comprising of collaborating partners from diverse and contrasting disciplines has been established by the group of scientific validation of principles of Ayurveda and its integration with genomics and modern medicine for preventive, predictive, and personalized medicine. She has more than 60 papers in prestigious peer-reviewed journals and has been over 100 presentations in national and international forum. She has been recipient of number of national awards, includes CSIR Young Scientist Award in 2001, DBT Young Woman Scientist Award in 2008, and most prestigious Santi Swarup Bhatnagar Award in 2010, Vasvik Award in 2016, and is a fellow of National Academy of Science too. And she is working with IGIB since I think 1997. It's been more than 22 years now. So, ma'am, I'm honored, and I'm in behalf of Srishti and my students, I welcome you. Over to you, ma'am. I'm sorry if I have missed anything. This is
no. It was quite a lot, in fact. so I don't know. Hello. Yeah. So I'll start.
Yes, ma'am. Over to you.
Okay, good afternoon, everybody. It's pleasure to be here. Pleasure to give a lecture to the students. It's always nice to interact with students. And if it had not been for the lockdown times, maybe you could have visited our institute. So this is our second campus of IGIB. We have 2 campuses in Delhi, and I am working out of this campus at present. So, what I'll talk about is the area of Ayurgenomics. But I think for the audience, before I get into Ayurgenomics, I want to get into the science of genomics and why we need to integrate Ayurveda and have this area called Ayurgenomics, and then get on to explaining, having got into this area, what are the kind of discoveries we can make, or how do we translate Ayurveda for contemporary sciences. Okay, so let's start very back in time when we know 100,000 years back, people started moving from Africa, and then they started colonizing in different time periods in different parts of the world. And when they started moving, they had to adapt to different kinds of conditions. For example, extremes of weathers, food sources, different kinds of pathogens. And pretty much humans over these 100,000 years have been able to colonize very many diverse geographies from high latitudes to arid regions to different high altitudes to different latitudes, etc. So all this would have not been possible just by being what you were in Africa, because when you move out, then these selection conditions might also have an impact on us, right? So and after that, when people also started moving, there also had been lots of shift in cultural practices. For example, in Africa, people were hunter-gatherers. Their dietary habits were very different from when people started farming. They started dairy farming. And when you do dairy farming, then the people would have to have ability to adjust to having milk, digest milk even till the later part of their life, right? So now it's seen that as these shifts in cultural practices happen, if people have to change with it, and if there are no, if there is an inability to change, then some kinds of disease can set in. For example, in populations of the world where dairy farming is not so prevalent, there would be large incidence of lactose intolerance, and that also happens to be in many different parts of the world. Whereas where the dairy farming has been very rampant, lots of people have actually got tolerated to lactose. So there's this very interesting book which I tell everybody in most of my talks, is that you people will have all heard about Charles Darwin's book, Survival of the Fittest. Now, this is a book which is called a Survival of the Sickest, which actually tells that how many of the conditions that we have, or which we think of as disease today, might have actually provided our ancestors certain kinds of advantages, but when they move to a different part of the world, then those very advantageous things can become maladaptive. So, so now, uh, as a consequence of all these kinds of cultural shifts, and also now with different kinds of lifestyles and all, when people study across the world, they find that the burden of diseases are also increasing, especially the chronic and complex diseases, which affect a large fraction of populations all across the world. And this is more for conditions like type 2 diabetes, fatty liver disease, whose incidence have actually increased in many parts of the world. And it once you have these complex diseases, it stays for a longer time. And if people are living longer, then most of the it has been seen that the last 10 years of their life they are not living healthily. So even if longevity has increased, it has been seen that in different parts of the world, the years of healthy life has reduced. That means you are living with diseases of chronic type. And if you are having these kinds of diseases, it actually requires medication over a longer period of time. So that not only increases the global burden of disease, but also the global cost of management of disease. Now, if we think about Indian population, and if we were to have a thought as to how this would impact our country, it is even more complicated, because India is a global melting pot of human diversity. There have been populations which have resided in our country at different time periods. There have been lots of movements from different parts of the world, and as a result of which Indian populations have all the world diversity in terms of languages being spoken, in terms of anthropometric criteria, and also we have our special marriage patterns, social practices, because of which we are also population within population, right? So all these would have actually made our population extremely diverse and different, and the health patterns are also likely to be impacting differently. So how do you study this? How do you go about understanding populations? Because clearly, if we do not understand the populations better, the disease patterns better, then we might not be able to have appropriate interventions for management of health and disease, right? So where do you see these patterns? How do you start to understand this, right? So to begin with, I will kind of ask you a question in the audience, that can everyone read all these lines? Anybody? Or can anybody read any of these lines? Can you just speak up?
Ma'am, I think presentation is not moving. This is only a first line.
Presentation is not moving. Oh, uh, okay.
Yes, now it's yeah.
Oh, okay. 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 So, so you did not get to see the previous slides. So I'll just breeze through it. So this is when I was talking about the world global burden of diseases, and also how lots of years of life are lost because of these lifestyle things like high body mass index, high blood pressure, sugar, and all. And this is the years of healthy life in different parts of the world. So as you can see, health longevity has reduced substantially in different parts of the world. And then this is the Indian population. So these are Indian populations, as we know, from different historical times, where people have inhabited India in different times of the periods, and also at what time different kinds of invasions have happened in the country. So as a result of which the population has become very diverse. And today's day, if you look at Indian population, then we have major, all the four major language groups which are spoken in different parts of the world are there in India. All the different kinds of uh, criteria of classifying people on the basis of morphological criteria, people are there in our country. Plus, we have a huge number of communities with intramarrying groups, which are called as endogamous groups, lots of languages. So that actually makes India extremely diverse. And from in this diverse population, if we want to have affordable health administered, then we will also need to worry about this diversity. So this is what I was just mentioning. And then if you want to understand diversity, from where do you start is where I came to. So I put some lines over here, and I want the audience, some of you might tell me that if anybody can read any of these languages, meaning any of these lines in their language.
I can read Gujarati, ma'am.
So what does it say, the Gujarati?
Tamil, ma'am.
The person who said he can read Gujarati, what did you read?
I don't think the line is grammatically accurate because "Ame te kari sako cho" means that you can do anything like that.
Okay, okay. Anybody else?
Ma'am, in Tamil, நாங்கள் அதை செய்ய முடியும்.
So what does this mean?
We can do it.
Okay. Anybody else?
తెలుగు మ్యామ్ మేము దీన్ని చేయవచ్చు.
Okay. So what
We
is
can do it.
Okay. So everybody is able to read one language, but nobody is able to read all the languages.
ಮ್ಯಾಮ್ ಇನ್ ಕನ್ನಡ ನಾವು ಮಾಡಬಹುದು.
Okay. So for everything, so this is a Google translation. Maybe that is why somebody said that you cannot actually read the. It's not grammatically correct, but since I don't know all the languages, I just used Google and I gave this English word and asked it to translate it into different languages. What I wanted to emphasize over here is that if all of us who look different are actually having molecules underlying which are all in different languages, then we would not be able to make sense of any of us, right? And that's where I wanted to get the idea of our genetic makeup. We are very lucky that all living organisms on earth have got the same unifying language, which is of the 4 nucleic acid AGCT. So if you actually take a bacteria, you take a plant, you take any animals, in all of them, the language of life which resides in our genetic material is this nucleic acid. And these are like the alphabets, right? And if you thread them, then they form DNA, and then these make sentences, which are the genes. And these genes are threaded into chromosomes, and the entire chromosome or the chapter can be bound into a book, and that is what is the genome. So every individual has all these letters, chemical letters, meaning their whole their makeup written in the sequence of these chemical letters, which are made of the same universal language of nucleic acid. But clearly, so the entire understanding of these letters to the function is what comprises the science of the genomes, and also called as genomics. So, some bit about the basic dogma. Many of you would know, or maybe all of you would know, that these DNA molecules, meaning which have these sentences with punctuation, are called as genes, and each of these genes can be read and converted to a message, which is by a method or process called transcription, to a message which is called as a mRNA. So this follows a chemical rule. So for example, if you have adenine, then you will have a uracil incorporated, you have cytosine, you have a guanine incorporated. So a DNA is read as a message, and this message in triplet is read as a codon for incorporation of a particular amino acid. So if you have an mRNA, that mRNA gets translated into a functioning moiety of called protein through this process of translation. And this translation happens also, or is dictated by what is the primary sequence. So if it is read in threes, then it will incorporate one of the 20 amino acid, and that way the sequence of the amino acid will determine what kind of protein would be, right? So in humans, there are nearly 22,000 different kinds of proteins, which are expressed differently in different parts of the organs, or different parts, different tissues, as a result of which you also give a tissue individuality, that what forms the brain, versus what forms the liver, it would be not the entire DNA is converted to entire set of protein in all tissue, but some tissues will have certain proteins and some tissues will have certain proteins, because of which you will get all these different and a particular organism built. So now you can realize that if there is a single change in the DNA, then that change will also be read as a slightly altered message. And then that message, when it gets converted into protein, could also alter the protein. So some of these changes might not do anything to the protein because it might be making something of the same kind, but some of them could actually become deleterious, right? So the genomics is all about trying to read the DNA and then trying to read the RNA. So when you read the DNA, it's a genomic component. When you read the RNA, it's a transcriptomic component. When you read the amino acid or the protein, it's a proteomic component. So these are multi-omic methods. But what is very interesting is because all the organisms share the same language, nearly all forms on Earth can be studied using the same set of platforms, even if you want to detect the DNA or the RNA or the protein. So that's what is the most appealing part of this science, because if it were all written in different languages, then we would not be able to make out any sense out of how related proteins could have maybe related functions. We could query that now. We can study a gene in human across organisms just because they would same protein can also share sequence complementarity, and there is a chance through genomics to understand that. So now the nucleotide sequence can change in different kinds of ways. So this is just an analogy. So there could be a substitution so that it makes another word, but that word does not make sense. So it could change to be able to survive. So there could be an insertion and it makes another word, but that word is very different from where it started. There could be other things. There could be deletions where the meaning has not changed or it has altered slightly. And these could then, during replication, have other kinds of things integrated. For example, you could have an insertion, you could have a substitution, you could have a deletion, you could actually have a substitution by a different letter, which could invert, then you could have fusion. So as a result of which, how you start and how you end could be very different. And that's how, when people have moved out of Africa, we moved with the same set of genome, but now you can very well make out who are the people who might be coming from European part of the country, who are the people who are coming from the African part of the country. We fundamentally share the same genome, but clearly there are differences in us, because of which what we eat, what we can digest, how we look, how we behave will become different. And that is all because when there is a change in DNA, it can change all through to the function, or it could also contribute to diseases. So some of the very drastic consequences of such kind of changes. So there could be one only change which could contribute to a total change in the shape of the RBC and lead to a condition like sickle cell anemia. So at the position 6, if glutamate gets changed to valine, 92% of the world population, this mutation is what is seen in sickle cell anemia, as a result of which the oxygen carrying capacity of the cell gets compromised, right? So this is one such kind of a disease, but there are 8,000 rare diseases all across the world, which also pose a bulk of the problem. But what we are also talking about is the common and complex diseases, many of which actually might have a genetic basis, but it also gets precipitated because of adverse environment or because of maybe other kinds of causes. So in order to make sense out of this, there was this major project initiated in the 80s, which was called as a Human Genome Project. The idea was that if we are able to sequence or get the sequence of the entire book of life of human, we might be able to use that as a reference to compare between people. And then we might be able to know if there is these kind of changes, then this contributes to these phenotypic differences or to diseases. So that was what was the Human Genome Project all about. We got the first draft of the Human Genome Project in 2001. It was nearly a 20 years of effort, because it was just not providing the sequence, but providing the technologies, how to read it, how to make sense out of it. Because if you just sequence it and get a sequence of AGCT, it will not make any sense unless you start to understand which part are sentences, which part do not make sense, which part makes protein, what kind of protein. So there's a whole lot of science behind the human genome sequencing. Now, from the human genome sequencing, the idea was that could we move to something by knowing the human genome sequence, we could actually have individualized medicine. For example, if we know a person's genetic makeup, would we be able to have that information useful when we are planning a baby? Because there are certain variations, certain mutations which run in the family. And if you are able to do a screening for carriers or screening at the prenatal stage, then you might avoid certain kinds of adverse outcomes. Or right after a baby is born, sometimes there are these inborn errors of metabolism which can be rectified at a much early age. So you could actually have certain variations used for testing those out. Or there is a whole space of undiagnosed disease, that there are some people who would get certain diseases later in life because they are predisposed to it. But is it possible to know about it much earlier, so that you can actually think about preventive mechanism? Or if you are differently susceptible to certain kinds of infectious diseases or other triggers, then could you avoid those situations? If somebody has cancer, there are lots of therapies, but many people respond differently to cancer therapies. So could you actually plan how to, what is the most appropriate therapy? Or even when you have therapy, sometimes lots of people have side effects. Some of them could be very drastic. So would the genetic variation tell that who are the people who are likely to have differences in therapy? And more importantly, how can you can increase the lifespan or the health span of a person by actually understanding your variations and how do they contribute to disease, and whether, if you know the impending cause of disease, could you actually have certain lifestyle adjustments so that you can increase the health span and also the cause of death, so that one can identify what are the major cause which could actually contribute to mortality. So this is all about the space of how genetics could change the entire span from pre-womb to tomb. So this is a very interesting article, and the whole thing is all about that. If you know about this, then you might be able to plan things much earlier. Now, this has led to this whole event of so whole initiative of precision medicine initiative. The idea is that once you know a genetic makeup of person, you can also start using different kinds of technologies to know in spatiotemporal dimension, what does he express in which part of the tissue at what time. So as a result of which, could you actually think about making a map of a person, just like you have a road map that you're going to some place and there is a danger or there is a roadblock and you take an alternative route. So if you're able to capture all this kind of information in spatio-temporal dimension, then you could you understand the pattern? Could you understand how you respond to a particular trigger, or how is your health trajectory? And as a result of which, would you be able to develop certain kinds of signatures which will tell you which are the people who need different kinds of intervention? So this is what is all what precision medicine is all about. And US and all have started this initiative in a big way, where they have recruited millions of people and they are capturing their data over a period of 10 years, and they want to use all these computational big data analytics to see what data they captured. From the data captured, could they identify certain kinds of patterns, and could they then use that information of those patterns to be able to get to predictive medicine? But before doing that, we actually, like I mentioned to you, we also need to contextualize that. It is not that if you have a static pattern, that pattern is likely to be same for an African to an Indian to a South Asian, because we are modeled by different population history. And these are all coming up as people are more and more studying genetic variation. For example, this is what is called as the root of human migration, or known as the root of human migration. And now what you can do is you can collect DNA samples from people from different parts of the world, and looking at the pattern of differences, you can start to see which populations are closer to each other. So this kind of an initiative, this was the human genome diversity, was conducted in populations all across the world. And each of these are one line of an individual who comes from Africa, from Middle East, or comes from Europe. And a similar color indicates that these populations are more closer to each other at the genetic level. So as you can see, if you look at the entire thing, then populations which come from the same continent are more closer to each other. And as the humans have moved, the genetic material also seems to be getting different in that way. So for example, populations of the Middle East are, after the African, are different from them, but Middle East also shares certain ancestry with the European population. They are different from South Central Asia. These populations are different from East Asia, with certain populations having both the components, and the Oceania and the America. So as people have moved, the genetics of people have also changed. And if we want to have suitable comparisons for knowing what changes contribute to what diseases, then one first needs also to understand that those changes are not just because we are coming from different population, but even if we are in the same population, we are different because of what kind of changes. So those are the kind of information. And then, as I mentioned, that people have moved to different parts of the world, have adapted different kinds of dietary habits. And then somebody started to study, let us compare people of populations who are on low starch diet and high starch diet. And it turns out that certain kinds of genes, for example, salivary amylase, which are used for digesting starch, there are copies which have of these genes which have increased in the population, which have got more starchy diet. That is because if you...
Hello, ma'am.
Hello, yeah.
Yes, ma'am, I have a question. What is this Bayaka and M-Beauty and Yakult in low starch diet?
These are populations. These are populations. These are names of the populations which have low starch diet. These are populations. So these are derived from these points. So there's this population called Biyaka. There's a population called Mabuti. So these are all different populations in different parts of the world, and the diet, what they have. And so some of the populations which are on high starch diet have more copies of the amylase gene. So that clearly tells that as people have evolved, their genetic their genetic changes also have happened, and some of these changes are because of the kind of diet you are in, right? Now what happens is that if you have more copies of a gene which allows you to digest starchy food, and if you're addicted to starchy food, then the same variation is also linked to obesity, right? So having this kind of an information allows you to know that what could have been an ancestral or adaptation for something which is because of a cultural transition could also then get to be contributing to disease susceptibility. So there is very interesting book called Food, Genes and Cultures, which actually talks about how people in different parts of the world, like you have Mediterranean diet, if the Mediterranean people have that diet versus if any other people have those diet, there are problems. And that is because of a special ability you will have to digest that much of olive oil or that much of different kinds of food, etc. So this book actually deals with lots of interesting stories about these genes and food and culture. And similarly, there are other kinds of things. For example, even different people have different kinds of taste perception. So even within a family, you might know that some people like sugar, some people like bitter, and these taste receptors have happened because we wanted to know what we are eating. For example, many of the poisonous food are bitter in tasting, and that is a protection for us to avoid such kind of toxic food, right? But that bitter is also there in other kinds of things which we might avoid. For example, many cruciferous vegetables are bitter in nature, but they are also rich source of antioxidant. So if you have a variation which allows you to taste bitter at a very low threshold, then you'll avoid such kind of food. Similarly, for different kinds of taste, you have different kinds of taste receptors. So even if you want to have a burger with different components, just like you go to Subway and ask for some kind of sandwiches, different taste receptors contribute to your kind of interest in how you would want to have the burger made. And all these in some way would actually connect to how in your body the microbes which are involved in digesting food will behave, and then how that is linked to your mind or your behavior. So many things are interconnected. So not only your genetics, but the genetic shapes what you eat, and what you eat gets decides what gets into your system, and then it also describes your behavior. So somewhere or the other, understanding of the genetic makeup has a lot of things to do with how you understand human individuality. So now, as I mentioned, so there could be a heritable component, there is an environmental component, and then there is an interventional component. That is, if you have a genetic variation which has something which you have inherited, you cannot do much about it. If you are residing in a particular environment, then also you have a particular makeup which allows you to adapt to that environment. But there is one component which is interventionable. That is, if you know that this is what is your response to a particular kind of diet or lifestyle, then that is an interventional component. So knowing all these kinds of breakup within you is what actually constitutes precision medicine, or what constitutes precision intervention. So I make it as an acronym of that you need to know who I am. So I is innate, am is acquired, and M is modifiable. So, but there are challenges. So this is not so trivial. Like I told you about 3 into 10 power 9 base pairs. But then if you think about the people of the world, then you will find that every person, there is a difference in 1 million, even between relations at 1 million places. So that can give rise to huge amount of combinatorial variability. So people have, after the Human Genome Project, have started to sequence more genomes from different parts of the world. And at the end, we find that there are 84 million places where variations can happen. So people are healthy, but they have variations. And these variations could be SNPs, or they could be indels, or could be structural variants, like I mentioned in the earlier analogy. And this can give rise to huge amount of combinatorial possibilities. People are now finding that there is a diversity in the kind of microbes which reside in our body, because there are 10 times more bacterial cells in our body which resides in, and different kinds of bacteria resides in different parts of the body. They would contribute to even more 100-fold order of genes. So human genome is not only 20,000 genes, but also the behavior of nearly 220 to 20,000 genes, which would be coming from the bacteria which resides in us. So if you now want to, from all of this, understand who is a normal human, it becomes a very challenging task. Because for us, what we started was that if we want to understand a healthy human, but understanding a healthy human means understanding all these variability. So mostly we know we are healthy if we don't have one particular kind of disease, but we really do not have a good definition of what is health, at least in the modern times, right? Unless we understand that, then we might not be able to accurately measure what is my disease state. Because if I have an average definition of health, then the disease state will also be an average definition, as a result of which how you kind of manage a disease is not likely to get personalized. right? It is very evident in an example which I would like to demonstrate, that everybody knows that nearly 92% of the people have this mutation which can cause sickle cell anemia. But a person with a sickle cell anemia can actually have different kinds of phenotypic outcomes. Some of the people can land up to stroke. Some people can have hemolytic crisis. Some people might have very painful syndrome. Some people can have chest syndrome. So you have the same mutation, but you have different kinds of outcome. And now people are finding that it is not only this mutation, but the presence of other kinds of mutation in other genes in an individual, in the presence of particular environment, can trigger particular kind of reaction which leads to particular disease. So the human is not only functioning as one gene, but it is actually functioning as an ecosystem, and you need to understand the whole thing in totality, that you have a mutation, what are the other kind of modifiers in the background of that mutation, so how likely you are to present with the same disease and in a particular environment, so that can you take care of that, and then how will your outcome be. So this is what is the challenge. And to be able to dissect this challenge, to be able to personalize medicine, then we have to kind of move from what we get from average studies to something which we want to personalize. But this is what is coming up by modern people. So many of you might have experienced that if in a house there are multiple people who have the same disease. For example, your grandparents will have diabetes, your parents will have diabetes. All of them do not share the same medicine, right? Everybody has their own dose of medicine calibrated according to their own time, so that even if a medicine has been found through a clinical trial to be effective, not all people respond to the same medicine in a similar manner. So this is something which is being done for very common kind of medicines. And you see these red colors and a blue color. It tells that this medicine, if you give to 5 people, it is effective only in 1 person. This medicine like omeprazole, if these many number of people are taking, only 1 person has an effect. So that clearly shows that even if you give the same medicine, people respond differently. And unless we know how much different they are, and can we then devise different medicines for different people, medicine we will not be able to kind of have a personalized approach, right? So now all over the world, people are saying that we need to now understand an individual, and then understand how are his medicine requirement, and then try to have that learning used for looking at another person who would actually share the same trajectory. So instead of doing trials on a lot of people, they are proposing 1 person trial, so that you observe one person, and then you aggregate that information to say what are the different kinds of patterns of people with different kinds of medicine. So so this is what is the conundrum.
Projection.
Hello, yeah. Anybody want ask something? Okay. So so there are a need for affordable solutions. So the idea is that can we, before having the medicine, have a way by which we can actually able to identify whether all the healthy are same, or there are patterns of differences in them. And if we understand that pattern, could we then segregate the people so that we are treating some people more effectively and not having a side effect of medication for the people who are not likely to be treated, right? So could you reduce on the number to get to a number where the medicine is most likely to work, and avoid giving that same medicine to a large fraction of people in which the outcome is not likely to be there. So this is where it comes in, this precision medicine thing, which actually in India, something like this has been practiced for nearly 5,000 years. And this has been recently acknowledged. We are doing this research. Dr. Bhavna is going to talk after me. She's a MD in Ayurveda, and we have developed this area of research for the last 20 years together. And the idea is of precision medicine actually is given in Ayurveda some 5,000 years back, and people are now acknowledging that what we are talking about precision medicine, India knew about it. In India, the concept of Ayurvedic medicine are laid out in sacred texts of Vedas. The goal of the medicine is to maintain balance between body, mind, and spirit, and treatment are tailored to each person's prakriti or constitution. So when I was telling you all about these tailoring of medicine and how people want to know the patterns, already in Ayurveda, there is a way to describe a person's constitution, and that is how today's Ayurveda doctors practice this medicine. So this is a shloka from Charak, and these are all documented. So what I am actually telling you, or what I am going to say, it is not that we are just saying it because of now, but it is something which actually has been laid down in the text long back. This is what we thought, that is there a way by which, if we understand how Ayurveda doctors understand human constitution, can we actually integrate it with the modern precision medicine and make or accelerate developments in this area of medicine. So we've been doing lots of interesting work in this area. So our idea was that Ayurveda talks about meaning modern medicine talks about all these things, but they use different, these things are not connected to each other. So you could diagnose a person for diabetes, let us say, but you will recommend everybody maybe to go easy on sugar, right? So the recommendations are very generic. So everybody will be asked to do lots of exercises. You really do not know whether everybody needs every kind of exercises, but all these recommendations are very generic, because there is a disconnect between what kind of genetic makeup is linked to what kind of diet, environment, and lifestyle. As opposed to this, in Ayurveda, this concept of Prakriti determines what is an ideal lifestyle, or what is an ideal environment, or what is a appropriate diet for a person. So once you know your Prakriti, then based on that, as opposed to symptomatic treatment, medicines are personalized on the basis of what your Prakriti is, and on the basis of your Prakriti, what your disease state is. So how does this underlying principle, meaning what is the basis of this, their diagnosis, or what is the underlying principle of Ayurveda? So this is something like how you have DNA as an underlying principle, or underlying unifying component for genomics, which will explain the transcriptome, the proteome component. Something like that is explained as a common organizing principle in Ayurveda, which is called as Tridosha. So according to this Ayurveda concept, every system will have three parts. One of them is called as Vat, the other is called as Pith, and the third is called as Kaph. Vat means the kinetic part, Pith means the metabolic part, and Kaph means the potential part. Now, if you take a biochemical pathway, in that you will build up something, then you will break down something, then you will transport something, right? So you have to have certain kinds of enzymes or certain kinds of genes functioning to build anabolism, something to metabolize, catabolism, then something to transport, and also so all these functions in Ayurveda are described in the context of all that governs the kinetic component is classified as Vat of the system. All that is governs metabolism is called as a Pith, and the potential component is called as a Kaph. So these are also described in Ayurveda. What are the functioning of Vat, Pith, and Kaph? When you describe Vat, that means what does Vat do, what does Pith do, what does Kaph do. So all these are actually from Sanskrit shloka, but you will see that these are also resonating with what we read in the biochemistry or molecular biology textbook. For example, for Vat, they say that it actually governs cell division, cell proliferation. So there are words, Sanskrit words for this, or it governs signaling, movement, transport, excretion, initiation. Pith governs digestion, metabolism, energy, host surveillance, and Kapha governs all that which builds up, for example, growth, storage, strength, lubrication, etc. So as you can see, every individual will have all these components, but if they have it in different proportions, that is what gives rise to different people having different proportions of Vata, Pitta, and Kapha, so different physiological or phenotypic attribute. So there could be individuals who would have more Vata in them, there could be individuals who would have more Pitta in them, or there could be individuals who would have more Kapha in them. So as a result of which, their inherent state of Prakriti is called as Vata type or Pitta type or Kapha type. So not every individual will have the same amount of these components. Depending on the proportion of these components, it will determine what kind of Prakriti the person is. And when there is a disease, then that disease is talked about in the context of deviation from that Prakriti or from that dosha. So for example, there is this Vata Prakriti type who have more of Vata, but if a Pitta Prakriti has that much of Vata, then that person becomes diseased, whereas the Vata person is still healthy. So how much is the deviation will actually be told only in the context of what is the baseline, and that is what an Ayurveda describes as Vikriti. So they assign people as what is their baseline state, and the disease is talked about as a perturbation of dosha from their baseline state, and all the drugs are administered to bring back the dosha to the individual's homeostatic state and not everybody to the same homeostatic state. So this is what is the underlying principle of dosha. And what is very interesting is, just like I mentioned, all the cells in the body don't express all the proteins. Similarly, in Ayurveda, it is described that all the tissues do not express all the doshas or the same kind of dosha. There are tissues in which more of certain kind of dosha is there. So there would be individual with an overall dosha and and a tissue with a particular proportion of dosha. So that would mean that for example, all these organs inherently have more Vata in them, all these organs have more Pitta in them, and all these organs have more Kapha in them. So if an individual is of more Vata type, then this organ might have more differentiating functions. These organs in Pitta type would have more differentiating functions because already their Pitta is elevated and these tissues are already inherently having more Pitta, so that a Pitta Prakriti person might have more differentiating features in terms of their appetite, in terms of their body features, in terms of pigmentation, all that is governed by Pitta. Or in a Kapha individual, there are particular organs. So this is how they actually connect an individual's dosha to an individual's phenotype. So what is done in Prakriti is looking at people with respect to how they move, how they walk, what is their physiology, what is their body frame, how they hear, what is their skin pigment, what is their appetite frequency. All these are assessed to assess what kind of Prakriti the person is likely to be. So that is why it is called as a unified principle. And that is why Ayurveda can actually have such a kind of very core underlying principle for personalized medicine. So according to Ayurveda, so when you look at Prakriti, there are a whole lot of features which are used to define a Prakriti of individual. So Dr. Bhavna, who is going to talk after this, she has over the last 20 years looked at nearly 20,000 people all across the country. She's developed a questionnaire which allows us to classify people into different kinds of Prakriti types. There are broadly 7 kinds of Prakriti types, out of which 3 of them are most extreme at the end of the phenotypic spectra, having more of Vata, more of Pitta, more of Kapha, or there are mixture with Vata, Pitta, Vata, Kapha, or Vata, Pitta, Kapha, or VPK. Now, what is interesting about this Prakriti is that the Prakriti of a person doesn't change from the birth birth to end of their life. So if you go to an Ayurveda doctor, they will, irrespective of whether you are changing, they will actually be able to assign your Prakriti, and that Prakriti doesn't change. So it actually is tells you what your baseline health is, and it also allows you to predict what are the likely responses you will have to particular kind of diet, particular kind of temperature, and what are the most diseases you might be more prone to depending on your Prakriti, and as a result of which how one can individualize medicine. So this is a review article which we wrote in 2015, if you people are interested in reading this further, because it actually has all these Sanskrit shlokas along with modern descriptions of all these things which are described in shloka. Even the modern people are talking about it, but they don't have such a cohesive definition of an individual's Prakriti. So for example, in Ayurveda, when they talk about Prakriti, then they tell that the Prakriti is governed by the genetic background of the person. Shukra, Shonit, these are what the genetic aspect is. Then there are things which are governed by the mother's diet and environment, what the mother is exposed to in the prenatal environment, which is called Matur, Ahar, Vihar, and all those things. And then it is governed by the background, Jati, Kul, what runs in the family, Desh, where you come from, what time you are examining, what age you are examining. All these things are taken into context when a person's Prakriti is defined. So this is a very amazing thing, because then what it allows them to also diagnose is that in different age, everybody will have their invariant Prakriti, but at different age, there are also these doshas change. For example, when you are born till 30 years, everybody would have a little bit more of kapha. But an individual who is of more kapha Prakriti will have more overt features. So he will have more propensity to have problems because of kapha perturbation. Whereas in the middle age, there will be more pith in the system. So people might have more diseases of pith Prakriti related, but people who are of pith Prakriti type might be much more having problems. Or for example, Vat Prakriti people will have more problems at the last stage because Vat increases as you age. Similarly, in different climates also, Vat Pitta curve fluctuates. And as you know, there are some people who have certain seasonal problems, and Ayurveda recommends particular kind of therapy for particular kind of people during particular seasons. For example, in springtime, cough increases in the system, but individuals who are of cough type might have more problems. In monsoon time, Vat increases, so Vat people might have more problems. So these are all the kind of perturbing condition which can be also assessed in an individual to see what is the Prakriti type and what is the nature of disturbance. And some amount of nature of disturbance can also be predicted by how the person starts off or what the person's baseline is. Similarly, at different times of the day, you are supposed to have a good meal during a time when your metabolism is more, and that is the pith time, or there is a time to rest, and that is the kapha time, and there is a time for body, all the movements to happen after you've eaten your food, and that is the vata time. So diagnosis also of these kinds of diseases are also made during taking into account the time of the day. So as you can see, what I was talking about, the GIS of human, Ayurveda already has this description enmeshed into the Prakriti, because you not only have a baseline constitution, but you also know the temporal or spatial dynamics of that person. So already the patterns which are wanting to be created, that kind of information already exists. And then there are diseases described, that who are the people who are likely to have what kind of diseases more predominantly. So if pith is more in the system, it could lead to skin bleeding disorders, or if vata is more in the system, it could lead to these kinds of abnormalities, or if kapha is more in the system, then it could lead to such kinds of diseases. So this is what Ayurveda talks about. And so the common principle of what we talk about as a dosha, it is used to diagnose health state or disease state based on what kind of features or phenotypes you look into, and that tells what is the cause, and accordingly you design a medicine. So this is what is the modern people looking into, network medicine. And here this description is already there, and one needs to but understand this in modern times to be able to port this information, because if we are able to do this, we would be much more ahead of time, because people are still groping with how to divide people. And here there is a methodology which actually maps the medicine, maps the drug, maps the diet, maps the lifestyle to a constitution. Such kind of information is not available. But then what do we need if we want to translate this? If we want to translate Ayurveda to modern way, then how do we evolve? How do we understand it in modern language? So first we want to have a framework which can port information. So whatever is told in Sanskrit or in Ayurveda, Charak or Sushruta, if I have to understand it in modern language, how do I interpret it? What is the way to query it? And when do I know that if I have been able to successfully integrate it, right? To an Ayurveda doctor, it is also a challenge that they see all the modern tests, modern parameters, how they contextualize it in their textual description, which are 6,000 years old. So for example, COVID pandemic we are seeing, and we think that it's a modern disease. But how would Ayurveda doctor see COVID? For them, they are also seeing diabetes, they are also seeing other kinds of disease, and for them, every disease is like a COVID, because they have to find an appropriate connect to Ayurveda to be able to treat them. But they have been treating for 5,000 years. So surely, if we are able to evolve a common language, then we might be able to get to have an integrative way of looking at Ayurveda. So now what is the challenge? So I'm just giving a small quiz to you to be able to say, how do we go about asking this question? So I have put something on the screen. And can anybody tell me that is there anything unifying to all these? Anybody in the audience? What is common between all these? Can anyone speak up? Hello, have I lost?
I'll start with A, letter A.
Okay, I'll start with A. That's okay. But then is there any other thing you can make out? No, nobody. Any other thing which you see common, or anything you can connect to?
Color of the text.
These are related with the 5 energy, the air, land, zero,
Energy.
absolute,
That's okay.
sky.
Colors.
Okay, somebody said color. All are colors. Acid is a color. Absolute zero. Okay, I can tell you all of them are color. So if you go to Google, you will find that if you give absolute zero and say color, it will show up this. If you say acid, it will show up this. But if I give you these colors, will you be able to get to this? The answer will be no, right? But if I want to make a shape with absolute zero, what is the information I would need? If I want to make any pattern out of this and say I want to make absolute zero and any shape, will you people will be able to make it by this only this much information? Hello, somebody speak up. Okay, I will go ahead. So many these ones, if you provide this kind of an information, only then you can actually make another shape. Is that right? So what have I done? I have converted this combination into a RGB code. And once I have that code, then I can actually make any pattern with the same color and it would be reproducible, right? So what I'm trying to say is that supposing we want to use information in Ayurveda, it is written in a particular language and we want to use it for a particular thing. If we want to do that, then we have to get it into a common unifying language to be able to port this information. And that is what is Ayurgenomics all about. That if I have to have a a whole interpretation of Ayurveda make sense to us and make sense to Ayurveda, we actually have to have such a kind of a translation which could be used by both the sets of people. So how do we go about doing it? So that's where I come in, that I gave you the genomic component, I gave you the Ayurveda component. Now, how do we go about integrating it? So this is what is the journey we started together, and Bhavna nearly 20 years back. So we said that there are these descriptions of Ayurveda, Vata, Pitta and Kapha, and Pitta is understood as metabolism for modern people. So, and Pitta is also prone to certain kinds of disorders, which are also kind of hemorrhagic disorders, if we go by the modern description. Vata is talked about as something which governs transport or kinetic component. So it could match to transport and signaling functions. There are proteins which are involved in signaling. And then Kapha is involved in storage. So there are lots of pathways which are known for storage. And then there are lots of diseases also known about obesity. So our question was that if we take individuals who have more of this, either Vata, Pitta or Kapha at the extreme, and look at their blood or look at their genome or look at their transcriptome, would they have more of these kinds of functions, and would then that be predictive about their predisposition to certain kinds of diseases? So this was what our working hypothesis was. So what we thought was we will take healthy individuals, but they should belong to only one genetic background and not to everybody. So that we ensured, because we were part of the Indian Genome Variation Consortia. So we take individuals of one genetic background, classify them using methods of Ayurveda to different Prakriti groups, and then take their peripheral blood and look at their DNA, RNA, and biochemical parameter, then synthesize the information and try to see what we get. Does it actually resonate with descriptions of Ayurveda? So this is what we did. So Indian population, we had made a genetic map of India. So we took people who are from this background, and from there we classified them into Prakriti types and identified who are the predominant Prakriti people, and then looked at whether they differ at the biochemical profiles or whether they differ at the expression profiles, and we started to see certain differences. This was our first paper. This was the first time we were able to show that whatever are described in Ayurveda could actually have molecular correlates, because we found that if we look at biochemical profiles, for example, lipid, cholesterol, and these are all healthy individuals, then we find that there are certain groups which have more of certain profiles. So you have a bell-shaped distribution of every biochemical parameter. So within that bell shape, Kapha individuals are more towards the higher side with respect to these lipid profiles, or Pitta people have more of the hematological parameters, or there are certain kinds of pathways which are linked to immune response, which are more in Pitta Prakriti, or olfactory transduction. So this was a very surprising observation, is less in Pitta Prakriti. Cell cycle is more in Vata Prakriti. So these are the kind of differences we saw in males and females. So we had done the analysis at the peripheral blood. And this is just a summary slide that we could actually start to see differences even within the healthy range. Vata, Pitta, Kapha had their own domain. So normally when we look at any biochemical parameter, if all of us are within this, we think we are healthy. But these people are differently healthy than people who would be in this domain. So that is what we could find, that even within the healthy, if we classify by Prakriti, then they are of different kinds. And that differences might make some differences to how they are predisposed, right? So this was our first paper where we actually showed there are differences if we classify people on the basis of Prakriti. And Bhavna could also get a shloka out of Sushruta Charak, where it actually showed that how Vata individuals, if they differ in cell proliferation, is what we observe. There are descriptions about how Vata governs cell proliferation, etc. This was about expression. Then we asked a question, whether these expression has a genetic basis or whether the genetic differences could also be visualized in Prakriti type, and whether those genetic differences could actually allow us to identify people who might be differently at risk of diseases. I'm cutting a whole story short because this has been a long journey, but I would get to one such discovery we made with respect to a gene, which is an oxygen sensor. So as you know, many of you might have gone to high altitude. If you go to low oxygen condition, then our body has to calibrate itself. So it has to make certain changes to be able to cope up with low oxygen condition, right? So there has to be a cellular trigger which tells that the body has to now cope up with low oxygen. So there is a gene in our genome which is an oxygen sensor, and if there is a low oxygen condition, that gene becomes inactive, as a result of which all the genes which are related to coping up with low oxygen get switched on. Whereas if oxygen is there, then so this is called as the EGLN1 gene. This is an oxygen sensor gene, and it binds to oxygen. So if it binds to oxygen, then it gets degraded, and all the so if you have a high oxygen, if you have a lower expression of this gene, then you will be able to adapt to low oxygen better. Okay. So this is a gene which allows you to calibrate in low oxygen condition. And what we found was that there were differences in people who were able to adapt, or who were having differences with respect to this gene. So for example, Pith Prakriti individuals had a variation in this gene, which allowed it to make less of this EGLN1, as a result of which by default it would cope up with low oxygen much better. Whereas Kaph inherently had high expression of this gene, as a result of which they would be coping up with oxygen much more poorer. This was the prediction which we could get out of the Prakriti based on their expression and based on the genetic makeup. Now we asked a question: if this differences is there amongst healthy people, then will that become relevant in places where there is low oxygen, for example, in high altitude? So what we found was that individuals who reside in high altitude had a genetic variation which was more like the Pith Prakriti. That means it inherently, because they are chronically in a low altitude, in a high altitude condition and a low hypoxic condition, they need to have a genetic change which would allow them to chronically adapt to that particular kind of condition. So we kind of asked a question: if that was so, if people from sea level go to high altitude condition and they are of this kind of type versus this kind of type, who are the ones who would be more adapted or more able to cope up with such condition? So we decided to test this out in a set of people who had gone to high altitude, and some of the people were not able to cope up with that low oxygen condition. And when we tested them out, we found that their genetic makeup were more like the Kaph kind. So this tells us that even before you go to high altitude, if you are able to maybe do the Prakriti of the people, or maybe do the genetic analysis of these people, you might be able to say which are the people who can do better at high altitude and which are the people who do not do so well. So this was the study which we conducted. And Ayurveda actually has descriptions in Sushruta, which talks about who are the people who are likely to be better adapted to very high altitude condition, and what are the kind of diseases which are more prevalent in high altitude. From here also, it actually becomes evident that it is likely that if you have more of Pith, then that is more adaptive at high altitude condition. Whereas if you are more of Kaph or Vata type, then you are likely to have more problems. So this is what we published, that for the first time, we could actually discover a gene which was linked to high altitude adaptation through analysis of something which was very unique, that is the constitution type. Recently, any such kind of a discovery is only valid if it is validated by different groups of population. 10 years later, the same gene has also been linked to high altitude in another altitude, that is Quechua, and this has also been linked to the aerobic capacity. So this clearly shows the merit of what Ayurveda talks about as Prakriti. And so to summarize this aspect, we've been able to filter out or distill the knowledge to classify people in a objective way, to be able to see what are the kind of genomic correlates we have. And from those genomic correlates, we have been able to discover things. One of the things which we discovered was the axis of low oxygen. So people will cope up with how they will cope up in low oxygen condition. It could be dependent on a prakriti of a person. Now, low oxygen is not only in high altitude, it could also be a case of many other conditions where if oxygen is compromised, then many diseases set in, and there are lots of therapy which are targeted to alleviating low oxygen condition so that people recover faster. So what does it tell? That if people are coping with low oxygen because of a genetic basis, then some of these diseases might not be so prevalent in those set of people. Or this also gives a lead to discovering things which could be relevant for targeting the oxygen pathway. So one of our colleagues, Dr. Anurag Agarwal, he could actually demonstrate the same hypothesis of ours, that even if you change oxygen through this axis, asthma, then there could be differential modulation of further outcome of inflammation and other kinds of injury. So, I will not go into further detail because it would be so very intensive now itself. So what we try to ask was, so we have a framework now that if you classify people by Prakriti, it's possible to interrogate them at the genomic level. And from there, we can make lots of discoveries which could have application or translational potential. So we decided to do this in different parts of the country, that how Prakriti in different parts of the country, would they have the same genetic basis? So this was a big initiative which was called as the Trisutra initiative, where we looked at different geographical region, tied up with lots of Ayurveda institutes, and tried to screen Prakriti, look at their different ways of looking at their different genomic profiles, and then we try to validate them in different kinds of functional things. So now just to end it, or just to get to the last part, how these kinds of information can become relevant in COVID times. So as you all are now aware that COVID has evolved in China, then it has moved to different parts of the world. It has evolved into different strains. And then in a different population, we are seeing heterogeneity with respect to the outcome, either because of a particular strain or it is because of particular population. So the outcome is because of population and also because of geography. So as a result of which, you might get many positive people who are asymptomatic, few people who get the disease, it is very morbid outcome, and then there are certain set of people who actually might be less severe in their outcome. So everybody who becomes positive is always worried, right? So the question is that, will we be able to use Prakriti to identify people who might have differences in outcome? And if we are able to do that, then could we actually have a stratified approach of dealing with them, either by modern medicine or by Prakriti? So the fact is that hypoxia response and hemostatic outcome is also being reported in COVID, that people require oxygen, people have thrombosis. And what we have shown is that different Prakriti type people have different oxygen responsiveness, hypoxia responsiveness, and also in different Prakriti people, the thrombotic or the bleeding outcome is also governed by genetic linkages. So this also was one part of a study which I did not get into, but it also says that not only Prakriti, in different Prakriti people, hypoxia response might be different, but what happens after hypoxia could also be governed by a person's Prakriti. And all these kinds of things are being seen in COVID outcome. So the idea is that could we actually use Prakriti to kind of differentiate people? And then there is this third aspect that we have found people who are of different Prakriti types. Hypoxia could be a trigger or could cause differences. Now in Ayurveda, if they treat Pitta Kapha, because here we have found that between Pitta and Kapha, there is a difference in hypoxia responsiveness. And if you modulate that, then so if you actually use a drug which is used in Ayurveda to treat Pitta and Kapha, would that actually modulate hypoxia outcome? And this is, I would leave to Bhavna to deal with, because she's discovered certain one such thing. And this we think has closed our entire framework, because if you are able to find such a kind of a thing, then in that case, if it is useful for one hypoxia condition, it could also be useful for another hypoxia condition. That way, you could repurpose whatever is described in Ayurveda to treat even diseases like COVID. So, and also there's this third part that Prakriti, whether only an Ayurveda doctor can do, or it could also be modern methods of AI and machine learning, can it be used to classify Prakriti? So could we actually implement it in a larger scale? So we have used machine learning methods to be able to use the questionnaire and show that if we classify people on the basis of the questionnaire which Bhavna has developed, and if we ask the machine to use the answers from the questionnaire, will that be able to classify people in an appropriate manner? And would those definitions, would those classification be also, so for example, these are basis of questionnaire, people were classified into 3 groups. When Ayurveda clinician overlaid their information, they actually got differentiated into Vata, Pitta and Kapha. But these are predominant Prakriti, right? So that tells that Prakriti has a phenotypic structure, and it is now possible to use engineering solutions to be able to convert this knowledge through all these methods to be able to implement them in a larger setting. So this was also one of the work which was modeled on the questionnaire which was developed by Bhavna. We could actually use machine learning to recapitulate Ayurveda constitution types. And it was a very interdisciplinary work where we had people from modern medicine to data to public health to modern doctors and Ayurveda doctors. So there are all kinds of authors in this paper. So this was also one very interesting work. And I will now just end. I just sort of have dealt with that how genomic has a need where Ayurveda has a principle which fulfills the need. And we've been able to provide a framework which allows us to query these. And if we are able to query, then there's a whole lot of information which we can port into our modern settings also, and have an integrative way of dealing with health. So we say Ayurgenomics is the new because it actually is precision medicine and precision nutrition also. It has advanced approach towards health management. It is contemporary. It not only can give us novel discoveries, because that EGLN1 had been discovered by our method in a very unique way. And before that, or just during that month only, the same gene got discovered, but with a very different kind of an approach. And this kind of discovery can actually bring about innovations in how we manage disease and wellness. And many of these we have also written up as a review article. So we've been working with a large consortia of people from different kinds of backgrounds, and primarily this has been our team. We have got PhD students who have been doctors and all. She's Bhavna, so she'll be giving the next talk. And we also have certain other kinds of collaborators from across the world and across the country. So thank you. I'll stop now. Hello.
Thank you, ma'am, for such a wonderful session, and it was really informative. And now, students, I request if you have any question and if you want to discuss with madam, now you have 5 or 10 minutes. Any questions, students?
Hello, ma'am.
Yeah, hello.
Good afternoon. I am Satyavrat. Actually, my question is, what are the different stages of translational research in between taking the findings to the community?
Different regions of translational research. So first of all, to be able to do translation, you need to have evidence, right? Can you just be specific about your question? Hello?
Yes, ma'am. Yes, ma'am. So suppose any traditional knowledge is there, and we want to take that traditional knowledge into Ayurvedic medicines. And what are the steps or stages you will follow to take that Ayurvedic medicines from the research state to the community?
Yeah, so that is what Dr. Bhavna, I think, will be dealing with it. So there are descriptions in Ayurveda. Now there are two routes to having the traditional medicine. Ayurveda people have already been practicing and giving the medicine. So people who have a belief in that system can already go to an Ayurveda doctor and avail the medicine, right? There would be people who might see that there is another person who is getting cured by a traditional medicine, but he would want to know what is happening, whether I will have a side effect or whether the medicine is really working. So what he will do, he will take a test report and say that if the modern benchmark is that if I'm able to get this value down to this level, then I know that your medicine is working. So you need a readout, you need a molecular readout. For example, if you want to avail medicine for diabetes, or if you say that a traditional medicine is working for diabetes, then you have to show it in a setting where people already define what is diabetes and people define what is cure of diabetes, right? So if you are able to get it to that setting, then only you'll be able to translate it. So to be able to do that, you will have to follow a series of steps. You will have to first take the medicine, test it out in model systems, which are used for testing the efficacy of drugs. Then you will have to take it to safety studies that it actually does not create any side effects. Then you have to take it to clinical trial where you test it on a limited set of people. Then you have to take it to a larger set of people. So the entire translation will have to start with the scientific evidence or a preclinical study to be able to take it to the entire last part. The other approach is that if somebody has done the science and you are able to interpret that science, then if you go to an Ayurveda doctor and he tells you he's giving you that medicine, will you be able to base your understanding and say, okay, I'll accept this? Because pretty much for modern people, if I go to a modern doctor with my ailment, he gives me some medicine, I do not ask him about science. I have it taken that it has actually gone through a process. You don't understand what the process is, but you take it depending on what that doctor is giving to you, right? So how do you do that? You only have the diagnosis that you have got this and you are cured of this. So there are steps, and what we are working on is how it works. If the principle, if the underlying principle on which they base all this medicine has a validity or has a way of querying it, then anything which is built on that, there is a possibility to query it, right? So that's what I would recommend as a translational approach.
Okay, ma'am. Thank you, ma'am.
Address your answer, address your question. Hello. Hello, Satyavrata, is that fine?
Yes, ma'am. Yes, ma'am. Got it, ma'am. Thank you.
Okay, okay. So,
any question now?
So did people not follow it?
That's what I was asking. So now I think I'm considering there is no question from student side. Students, if you have any question, you can drop down or you can mail me. I'll convey that to the madam. And thank you so much for your time, ma'am. It's really wonderful and informative. And this topic is actually new for many of us. So maybe that was that may be the reason behind this.
Sure, sure.
Yeah.
Sure.
Thank you so much, ma'am. We look forward for the future collaboration in such programs.
Okay, okay, bye-bye.
Thank you. So students, now we have next session at 2:30, but we'll meet at 2:25. Without fail, please join. I know you have very short break, but take your lunch and we'll meet at 2:25. Is that fine?
Okay, ma'am.
Yes.
Okay. If you have any questions regarding the earlier sessions, you can mail me or you can ask me in the WhatsApp group, and we'll let you know later. So then see you at 2:25.
Okay, ma'am. Thank you.
Okay, ma'am.
Okay, ma'am.
Neen Gandhi.